In early discovery, LCMS gets you answers fast mass, retention, and a quick purity read. But sometimes the data raises a bigger question. Is the structure truly what we think it is? Or are we looking at an isomer, an impurity, or a degradation product? When do you need to dig deeper into structural elucidation? Let's explore. LCMS is excellent for supporting identity assignment, but mass alone does not prove structure. Different structures can share the same molecular weight, and sometimes even similar MS behavior. This ambiguity can be a real problem. So, when do you escalate to more advanced techniques? The key is to be decision-driven. If the structural uncertainty could distort your structure-activity relationships, mislead your potency interpretation, or affect which candidate you select, then escalation is absolutely justified. It's all about making sure your data is leading you in the right direction. One of the first steps in escalation is tandem mass spectrometry. This technique provides fragmentation patterns that can support your structural hypotheses. It's especially useful when you compare the fragments to expected substructures, known analogs, or reference standards. Think of it like a structural fingerprint that gives you more confidence than just a mass value. But even tandem mass spectrometry MS has its limits, especially when you encounter the isomer problem. Isomers can share the same exact mass, and sometimes even similar fragmentation patterns, making them incredibly difficult to distinguish. That's when you need orthogonal evidence, better chromatographic separation, specialized Tyrrell methods, or the gold standard NMR. To get closer to a definitive answer, high-resolution mass spectrometry, or HRMS, is a powerful tool. HRMS provides an accurate mass measurement that narrows down the possibilities to just a few plausible molecular formulas. When you combine this accurate mass with the isotopic pattern and MSM's fragmentation data, you can substantially increase your confidence in the assigned structure. HRMS is most valuable when you see unexpected peaks, suspect modifications on your molecule, or are working with completely new chemical scaffold situations, where you need much higher confidence than a simple nominal mass can provide. But when you need the strongest possible confirmation, nothing beats NMR. In discovery, you don't always need a full, exhaustive characterization package. Instead, targeted NMR experiments can resolve key structural uncertainties quickly and efficiently. So when is NMR really worth the time and effort? It becomes essential when biological activity is inconsistent from batch to batch, when isomers are a plausible explanation for your data, when you're about to select a lead series for further optimization, or when you need high confidence before investing in a scale-up synthesis or critical in vivo studies. Sometimes, the most important molecule in your sample isn't your target compound. If an impurity or a degradant might be the real driver of the biological activity you're seeing, you need to isolate it. Rapid isolation using techniques like preparative LC or targeted fraction collection can provide enough material for a direct MSMS comparison or, ideally, for NMR confirmation. This helps you understand what's really happening in your assay. However, it's crucial to avoid over-elucidation. Discovery analytics should not become a full-blown impurity investigation program. Chasing down every minor peak is a waste of resources, unless that peak is directly affecting your scientific decisions or the interpretation of your results. Knowing when to ask for help is also key. Some structural questions benefit from specialist support. Don't hesitate to pull in spectroscopy experts, metabolite identification scientists, or separation gurus. Their expertise can accelerate the process and ensure you get the correct interpretation, saving you time and preventing you from going down the wrong path. So, what are some common real-world triggers for escalation? Think about unexpected shifts in potency, batch-to-batch, inconsistencies in your assay results, the appearance of new degradation peaks over time, or changes in your synthetic route that might introduce new byproducts. These are all red flags that demand a closer look. It's important to remember that this isn't about ICH-style impurity identification. In discovery, structural elucidation is about reducing scientific uncertainty that could mislead your project. The formal regulatory expectations for impurity characterization come much later, when a compound transitions toward clinical development. In the early stages, it's all about making informed decisions to keep your science on track. Used strategically, structural elucidation protects the integrity of your SAR interpretation and keeps your research programs moving forward efficiently. The takeaway is simple. Go deeper when the answer changes the decision, and stay lean and fast when it doesn't. Thanks for watching. In our next episode, we'll dive into analytical risk and rapid SAR cycles, and explore how even small errors can significantly distort your outcomes. Make sure to subscribe so you don't miss it.